So with gene transfer, we talked about the idea of horizontal gene transfer last week in the evolutionary process. Now I wanted to get into the mechanics of it, like how does that actually happen? And there's actually several ways. I'm going to cover some of the key ones, and then as we move into some of the other topics, you'll see that there's some other ideas out there that have just been recently discovered. But transformation is one of the earliest ones that was discovered. This is the idea that you have, they actually call it naked DNA, which just means it's DNA that has been expelled, not really. So if you take a cell and you break it open, the DNA comes pouring out, and then it's just out in the environment. It's free and naked, okay? And so that's what you're seeing here. They had, I believe, E. coli that they caused E. coli to burst. And all of that stringy stuff is the DNA. So DNA in most bacteria is circular, big circular piece of DNA for the chromosome. Okay. There are a few bacteria that have linear DNA for their chromosome, but most is circular. And this you can see is circular and twisted and big. So now a nearby microbe that's still living can take up some of this. This is a lot. They wouldn't like take the whole thing. That's a lot. And so what tends to happen is there'll be chunks of the DNA that break, and microbes can take up those pieces quite readily. And that's what we see happen. So about 10 kilobase pairs, which is still a decent chunk, can get taken up. And a single neighboring cell, maybe they'll just take up a chunk, maybe they'll take up several fragments. It just depends. Now, they do a lot of this, of course, in the lab where they manipulate things and they're studying things. And so they're extrapolating. They're saying, well, we can get it to happen in the lab, so it's probably happening something like this out in real life when the microbe is living its life on its own. Okay, so now sometimes they can get the microbe when they're working with it in the lab. Sometimes they can get the microbe to easily do this. Other times, not so much. And a lot of it has to do with just understanding how to work with that microbe. But basically what they say is, yeah, it seems like not all cells are transformable, like they can't get it to happen. Now, does that mean it never happens? Maybe we just don't know how to work with the microbe and make it happy enough to do this transformation process. But they have gotten down to the DNA level, and they said, well, it does seem like you need some key genes in order for this to happen, for the microbe to take up this naked DNA. And so they finally got into that level of research, and they were able to see that there does seem to be these special proteins that are necessary for the microbe to effectively take up the DNA. Now, some microbes will produce these protein complexes, not all, but some, and they're called transformosomes. So I would like you to know that term. And it's just a protein complex. I'm not going to get into huge detail on it, but basically the model organism that they study for a lot of this idea is a Bacillus species, B. subtilis, which you'll actually work with tomorrow in lab. And what they see is that there often needs to be this communication process that's called quorum sensing, which we'll get into that more as the semester goes on. But quorum sensing is a way that microbes assess what's going on around them. They can use quorum sensing to communicate with other nearby cells. They can use quorum sensing to assess population levels around themselves, which can be very, very important. And so we'll talk about quorum sensing when we look at some of the microbial ecology and how microbes end up communicating with each other or with other organisms. Okay, so the idea here is that they can get it to happen in the research lab. Okay, we see microbes taking up DNA. But then the question is, okay, they can do it, so why would they do it? So I think, we've already hit upon the idea that they might want some of this DNA because it might contain genes, right, that could help the bacteria and make them to better adapt to their habitat. So certainly they might take up the DNA and then incorporate it into their own chromosome and then express it. And hopefully it would give them an adaptive advantage. However, if you look at DNA, there's also a potential option that they might just use it for food because it's got nitrogen and phosphorus. I mean, it's got some nutritional value if you degrade it and use it. So that might be an option too. So that's something that they definitely think could be going on. And maybe it's to repair. Maybe that chunk of DNA somehow will help them repair a damage to their own DNA. So all of this has been explored, and it seems like these are all likely scenarios, but the one that really grabbed the scientists' attention was the horizontal gene transfer, because then those genes get expressed and can lead to better adaptations. It might even lead to speciation over time. So this has been something that they've been looking at pretty carefully. So with transformosomes, I said some do that. Not all will build these protein complexes. So here is an example of a transformosome complex. Now, this is a bacteria that is gram-positive. For those of you, did anyone take micro at all? Oh, yeah, fair bit. So if you've taken micro already, you know about the gram-positive and gram-negative types of bacteria. And you're certainly going to learn about that in this class too. And so the gram-positive versus gram-negative has to do with what kind of cell wall they have, because not all bacteria have the same type of cell wall. So anyway, so we often see gram-positives do certain things certain ways and gram-negatives do it a little differently. Here we have the gram-positive, we'll get into that later, but basically this looks really busy, right? I mean, there's a lot going on here. But what I want you to key in on is the transformosome. So this kind of lime green blob here in the cell membrane, cell wall area, that's the protein complex that will be used to bring in the naked DNA. You're seeing that it's not for free. Looks like there's ATP being used. So if they choose to do this, there's a cost, energy cost. And then all this other stuff that you see going on here, where you see these CF, and then it's stocking with a protein here, and then you see this arrow, and these are different genes. I'll come back to that, right? We're not going to get into that right now, but I'll show you this picture again when we get into quorum sensing. Because all of this is essentially part of the quorum sensing concept. And so, like I said in the previous slide, quorum sensing is involved. This is it, but you don't need to know any details yet. Just know that, yes, they are doing the quorum sensing. And then the transformosome complex. And what you're seeing here is, fragments of DNA coming into the cell. So lots going on there, but for this section, you just need to know the basics. Okay. All right. So it seems like a lot of gram-positive bacteria do this kind of thing where they build this transformosome. So all of these genes are getting turned on. They're trying to say that, all of this is needed to build this transformosome. Okay. Now with the gram-negative, it's a little bit more straightforward. They don't use these proteins called competence factors. It's a little bit more straightforward, where you have no quorum sensing, doesn't seem to really be factoring into it in any way. And it seems like, at least in the lab, when they're working with them, they're either competent or you can make them competent by starving them. And so you stress the bacteria out, and that switches into their behavior of, I guess, I'm getting starved. It'd be kind of a good idea maybe to access some DNA. That might help my situation here. And so you're kind of pushing them into taking up some DNA. Now, they don't build a transformosome complex. They do this funky thing where they grow a pilus, which is this long chain of proteins. It's just protein subunits put together to make this long, thin protein strand. Hair-like, really thin. And then they use like the tip of the pilus to like snake some DNA and then drag it into the cell. It's really kind of funky. And they have to make it through the cell wall area, cell membrane area. So I'm making it, there's more to it than what I'm telling you. So it's a little complicated once they get the cell wall area, but they manage it.
What do you mean by competent?
Competent means, are they able to take up the DNA? And you'll hear that if you work in a lab where they are genetically modifying a bacteria, let's say, we have to make the cells competent. Which means you have to manipulate the conditions so that they're very capable of taking up DNA to make it more successful, to get the genes in that you want to get in. So here's kind of an image of it where this is already retracting. So all this green blobs, those are the protein subunits that make the pilus. And usually that's outside of the cell, and then they can kind of retract it. And you can see here the tip of the pilus snakes some DNA, and now they're retracting it so that the DNA comes into the cell. And that's really all you need to know about what's on this image. All right, we're not getting into the details. Just know that they use a pilus structure and they snag it and drag it. Okay, so transformation is just uptake of free DNA. So if you have a population of microbes all hanging out in close physical proximity and some of them lice and break open, the DNA spills out, starts to break down, neighboring cells can take it up. There's another method called conjugation. Do need you to know that term. So conjugation always makes me think about conjugating verbs I took different language classes, but that's not what we have going on here. So now what you see are two cells. Can you see that yellow thing in between? Okay. So that is another type of pilus. Okay. So there's different kinds. The plural of pilus is pili, P-I-L-I. There's different kinds of pili used for different things. If you've taken Med Micro, I'm sure Dr. Sharp talked to you about some of these really funky pilus structures that can make pathogens very pathogenic. But what we're talking about in this case is it's a way for two neighboring cells to make physical contact through the pilus, serving like a bridge or a tunnel. It's really a tunnel if you think about it. What this is again, it's protein subunits that are formed, but it makes a tunnel. It's hollow in the middle. All right, so it's a tunnel that goes between two neighboring cells. This one's pretty impressive. Doesn't that look nice and long? I mean, that's quite impressive. So what happens is that's the physical contact that we're talking about. At least that's the initiation step. And then what happens is, so let's say this bottom cell through this pilus synthesized it, got it, and then it snagged into this cell. Now what's going to happen is this one will actually tug this cell close to itself and get so that they actually butt up so that there's physical, even a tighter connection than just the pilus. And so they actually refer to it as a sex pilus. You'll see that in the literature. They'll say it's a sex pilus. And the cell that creates it is called the donor cell. Okay. So now what we see happening is with the help of the pilus, you'll get transfer of DNA from the donor to the recipient. And it usually requires these special plasmids that are, they have the right genes on them that allow the plasmids to be transferred. Okay. So these plasmids basically are like one-stop shopping. It has all the information on it necessary to build the pilus, plus allow for the transfer of part of that plasmid. So now you see a mock-up of 2 cells and not quite as dramatic of a length of pilus there, but you're seeing that there is this, they're trying to show that tunnel or the pilus structure. Now, they didn't go into details on this drawing, so typically what they've done most of this work at E. coli. By the way, E. coli, Escherichia coli, which you'll work with quite frequently in lab, it is kind of like the guinea pig of bacteria. So if they gotta pick a bacteria to work as, to see what's gonna happen in the lab, they usually pick E. coli. But often the Bacillus species too, and E. coli is gram negative. But anyways, so here you have a donor cell, which just by convention, we call it the F plus cell because it's the one that has the plasmid. And then the recipient cell is F minus, meaning it doesn't have the plasmid, not yet, okay? And so in this case, what we see is here's kind of a step-by-step process of conjugation. And again, it's going into more detail than what I expect. So just listen up here. So what I want you to know is that, yes, the donor builds the sex pilus and it hooks on to the recipient. And then you see how it's retracted and kind of brought the recipient close and then very close here. And you can see that you have a nice tunnel system now with the transfer of DNA now initiated through that. And the thing to pick up here. Oh, that's interesting. So the way they drew it, the plasmid is right here, that F factor that they're calling, that's the plasmid. And then the thing to the left of it is the chromosome. All right, so what they're showing here now that the transfer is initiated, they're trying to show that just one strand of the plasmid. Remember, plasmids are small circular pieces of DNA. They're double-stranded. And so you just see one strand coming over. And now that image right here, you see the strand is fully over, and they're trying to show that now it's making a complementary copy of that strand. Right? And now the very last image, you're seeing the recipient has made a full complementary copy. So it's got the plasmid, good. But the donor didn't lose the plasmid. It still had one strand left over. And then it made its own complementary copy. So think about it. You're a bacteria living in a habitat with neighbors. And let's say a bunch of toluene gets poured onto that soil surface where you live. And you happen to have a plasmid that has a gene that can degrade toluene. And you're feeling generous today. So you're like, well, I can share that plasmid. Would you share the whole plasmid and purge the whole plasmid out of you? No. You keep back some. It's like, I want to survive the toluene too. So I'll give you half. And you can make a copy. I'll keep my half. I'll make a copy. So we're up to the double-stranded again. So really, if you think about it in an ecological context, it makes a lot of sense that they only transfer one strand over, not the whole thing. Because then you lost your ability to degrade toluene. That makes sense? Did you have a question?
Does it matter if it's the outside or the inside part of the plasmid?
No, you don't need to know that. I don't even know, to be honest, which part gets transferred really in. So, no. And like, there's a lot of words on here. You don't need to get into the details. It's just the sex pilus idea, getting the cells close, you create a tunnel that one strand goes through. But one strand, think about it from an ecological context. You would not want to just give out your whole plasmid because there's a lot of genes on there that may be super useful. In the case I provided, it was toluene. You can survive toluene instead of being killed by it. Now, every once in a while, in the lab when they were working with these E. coli and other bacteria and seeing how this worked. Sometimes those plasmids just stay as plasmids, separate from the chromosome, but every once in a while, they would see the chromosome would paste in, the plasmid would paste into the chromosome. So if that's the case, then they would refer to that as the HFR type plasmid. It's just a way so they can talk about it. So when someone says, oh, look at that HFR plasmid, they are saying it has pasted in, the plasmid's pasted into the chromosome. Now it still functions, you can still get gene expression and it's still functioning, but it just happened to paste in. One interesting thing though, a consequence of this is that when they let that bacteria keep going and they're like, okay, let's keep growing it with some neighbor bacteria in there. It's usually done in a broth media, so they're, swimming around, interacting. And what they see is that plasmid that's in the chromosome will start to get donated out to a neighbor. And sometimes some of the chromosome DNA comes with it. And so you have this interesting idea where it's not always just the plasmid, a strand of the plasmid that will get transferred. It'll be some of the plasmid plus some of the chromosome. And again, so now that's a way for the recipient to get some interesting genes that it might never have had before, coming off the chromosome from its neighbor. Now, in the research lab, they can force the situation where they can make the whole chromosome go over. Now, again, if you're a living microbe, you wouldn't do that, right? You wouldn't give up your chromosome, you'd die. But experimentally, if you're curious, it takes about 100 minutes for the whole chromosome. But that, I'm sure, really doesn't happen in a natural setting. All right, so again, this is just a visual. We are not going to get into details of this. I'm just trying to show you the purple is the plasmid. And what they're trying to show is that it got pasted into the chromosome. And then if you keep that microbe up and running, it may encounter the right conditions where that strand can get mobilized again and like cut out of the chromosome and sent over. But they may take some of the flanking pieces of chromosome with it and get it over into the recipient. So that's kind of an interesting little twist to the process. And then, so here, this is the part 2 where you can get part of that plasmid, like I said, that can get cut out from the chromosome, and it might contain some chromosomal genes coming with it, some of the flanking. And so that could be good for the recipient. It could give that recipient cell some extra genes to see if it works and provides a good adaptive ability. So the more they looked into it, the more funky it was getting. So here's part 2 again, where they're trying to show like, okay, once it's in the chromosome, then it's going to get cut out under the right conditions, and it might take, so this gray and yellow and red, those are the flanking pieces of DNA that are coming with it, okay? So what I've been talking about are like 2 microbes hanging out with each other, and they're capable of doing this. One of the other fantastic discoveries that was made was the idea that you can get gene transfer between domains of organisms. And when they saw that that was happening, it was mind-blowing. So have you ever, there's several of these in town. You don't even have to go out into the woods. Have you noticed trees that have these big tumors? We call them galls, G-A-L-L, in the botany world. This is called crown gall disease. It's caused by a bacteria, and the bacteria is using conjugation to do this. So here in the lower image is the surface of a plant cell, and you can see all these pathogens. They're about to gain entry into the plant cell. So this is a pathogen that's called Agrobacterium tumefaciens. I would like you to memorize that species, please, because this is historic. I mean, this is like a big deal. And so Agrobacterium tumefaciens, the plant pathologists who are studying this pathogen, trying to figure out how it causes crown gall. And it can cause crown gall on many, many different plants, not just trees, but it's pretty noticeable on a tree, but it can happen on herbaceous species, species that are grown for food production, for example. And what it does is it has a TI plasmid. The T stands for tumor, which should have been G for gall, but you know, anyways, they named it TI. The I stands for inducing. So this is a plasmid that has the genes on it necessary to create this gall. Now you might say, why? Why would a bacteria have genes that can allow for this to happen? So let's think about that as we work through this a little bit. So what happens is that there's conjugation that occurs between the bacteria and the plant cell with a sex pelvis idea. And then the plasmid from the bacteria, one strand of it, one strand gets transferred over. And it incorporates into the plant chromosome, the plant DNA, the chromosomes. And it has genes on it, several genes on it. One is to get the cell to do localized cell growth. So it contains a hormone, a gene that codes for a plant hormone. And it makes the plant overproduce, like just go through lots of cell division without any differentiation, which is like a tumor, you know, like a cancer tumor, that's essentially that. But this isn't causing cancer, okay? But it is this, mass of kind of undifferentiated cells. And if you were to like slice into this and look at a thin section under the microscope, you would see it's chock full of bacteria. So basically the bacteria say, here's my plasmid, express it. One of the genes is a hormone. I want you to make a bunch of cell division, create this gall or tumor so I can live inside it, have a cozy home. So now they're well protected. The bacteria are in there. Then the other, there's some other key genes on the plasmid that tell the plant, basically the bacteria says, make a home for me. Next, start expressing these genes that make a nitrogen containing compound that the bacteria use for nutrition, but the plant can't. Isn't that just, so the bacteria is like, make this nutrient, and by the way, you can't use it because you don't have the right enzymes for it, but I can. So it makes the plant make a nutrient that the bacteria can use. So that's pretty good. And it rarely causes the plant to die, which again, from the bacterial's perspective, that's pretty rational, right? I mean, if you were going to infect a plant, and you have this cozy home, would you want to kill it? You probably want to just keep going. Like, I'll just live with you and you can keep feeding me and you built this nice home for me. I'm not going to kill you. So this disease really doesn't cause death, except if the plant might be already stressed with some other disease, then this might tip it over into, it can't survive it. But that's why you see so many trees around here with these big growths, and you're like, well, every year it creates new leaves and it's happy and it's growing. And it's because they're kind of coexisting with each other, under those conditions.
Thanks for the fungus too.
Well, so this gall is caused by Agrobacterium tumefaciens. There are other plant galls that can be caused by fungi. Often they're like tiny little galls that form on leaf surfaces. You'll see like little bumps that look like little tiny tumors. There's even insects that will, there's certain wasps in their venom. They have a plant hormone that causes overgrowth, and they land on a branch and they'll sting the branch. And the plant cells get that dose, that flood of hormone, and it causes them to create like a gall, and then that's their nest. So insects can kind of take advantage of this too, like create a home for me, and they just kind of repeatedly sting the branch. Kind of crazy. Yes,
What's the relationship between like donor and recipient cells and conjugation? Does it vary between like parasitic and symbiotic?
I think it's the same process, kind of a universal process in how they do it. But it really does depend what plasmid gets transferred, what's gonna happen next. You know, like in this case, the plasmid had the right genes to create the gall and then make that nutrient for that, the bacteria. Yeah. So there's different plasmids. If any of you have taken BI 218, is that the intro to cell? Like when they buy plasmids that you might have worked with, you can look that up, you know, a scientific company, and they have pages and pages, you know, of different plasmids that are out there and available. And there's, well, here, let me just show you on the next. So here is a partial description of different plasmids. And so you can see that there's a bunch of different bacterial species. Sometimes they just say wide range. And then you can see the different phenotypes that if those genes get expressed, what is it doing? And so here you can see conjugation, the actual process of getting conjugation to happen, yeah. Antibiotic production, that's something that I'm very interested in because I work with Streptomyces bacteria a lot. And I want them to produce antibiotics because that's how they kill off plant pathogens primarily. And so we know that many of the plasmids that Streptomyces have they have genes that code for antibiotic production. And so kind of the flip side of that is resistance. So that's why this should concern all of us, this process of conjugation, because it can allow for the spread of genes on plasmids from one donor to recipient that can make the recipient resistant to antibiotics. So from our perspective, not a good thing. And actually, it's a very serious process. If you talk to medical microbiologists at the last conference I went to, it's like, after you have that conversation, it's like, where's the nearest bar? I'd like a beer, please, you know, because it's scary. And they're working very hard to try to come at this from different angles of research. How do we minimize antibiotic resistance? Because it's a problem. There's some superbugs out there, some pathogens that are resistant to all known antibiotics that we have. And so if you get sick with that, hope you have a good immune system. So, and yeah, so on that happy note, then there's other things like virulence, so pathogens, they get their ability to become pathogenic, often through plasmids. And what else? Oh, and then degradation. So this is a positive thing. Many microbes, if they get some of these plasmids, it allows them to survive, like the presence of toluene and benzene and camphor, and it actually allows them to break down these toxins and use them for nutrition. So if you get those plasmids and you're in a contaminated area, which are in a lot of areas out there, that would allow you to survive. And not only that, but break down the chemical for your food, which then helps us because it breaks down the nasty chemical and makes that area less toxic. So lots and lots of plasmids out there that have been studied. You can buy these from scientific companies and you have the bacteria to thank for it. That's where they're getting these from. So if you acquire a strand of that plasmid and you make your complementary coffee, you get it up and running, some of those genes can lead to better adaptation. It changes your phenotype, so how you behave, and it could give you an advantage. So if you are a recipient, that's a great thing. They refer to this process of conjugation as infectious. They're trying to get across the point that this is pretty easy for bacteria to do. It's pretty straightforward. And that's why it's so scary when you think about antibiotic resistance. And now we know it can be done through very distantly related organisms like Agrobacterium in a plant cell. So that's pretty wild. And certainly between bacteria and fungi, it's known too. So when Agrobacterium was discovered that it could transfer its plasmid into plant chromosomes, by the way, that was a huge thing for molecular biology research, because if you try to genetically transform a plant, it's really hard. Because plants have really tough cell walls. Trying to get DNA through that, it's really hard. It's a complicated process and with low success rate. If you can just have a bacteria do it for you, oh, there went hours and hours of effort, all of a sudden it's super easy. And so that was a huge boon to the molecular biology research in how to get foreign DNA into plants. That was a big, like pivotal moment in the history of that kind of research. This is another table from a different resource, just showing, I like that they break it up into like, you have archaea transferring to bacteria, bacteria to bacteria, bacteria to eukaryote. So they're giving some examples of this. You can get this transferred a strand of the plasmid to organisms that aren't really closely related. It's pretty phenomenal. So the antibiotic resistance is a big concern, but they can use this idea too, though, for good things, like it's, breaking down toxins and things like that. With the streptomyces that I worked with, I had a problem because the antibiotic that was killing off the plant pathogens I was working with was on the plasmid. So remember what I was saying about flagella and lab bacteria? So if you're a baby, so the streptomyces I worked with, I just had them in the lab and I worked with them and grew them up in the lab. And they had the sweet life. What do you think it might do with those plasmids? Ignore them, burn them off, purge them. That's what happened to my streptomyces. They got so comfortable living in a lab where I gave them everything they needed. They just stopped producing antibiotics. I was half a year away from graduating and my bacteria stopped doing what they had to do. Talk about panic. Oh my goodness. Panic. However, luckily, if you work with microbes in research, freeze them, get them pure and freeze them. And I did that early on during year 1. So as I'm, getting near the finish goal and my bacteria stopped working because they purged their plasmids, I went back to the frozen stock. They still had it. Thankfully, oh my gosh, was I relieved. So just be careful if you work with them in research, you should have a good protocol for preserving them. So that, because they can get the sweet life and just decide I'm done. I'm not gonna do that anymore. Well, with the streptomyces, I had some frozen 20 years. And then made it, but they form spores. So they're a little bit better. I get nervous if all of, like, let's say I have one specific bacteria that I really want to keep working with. If it's been frozen for longer than 3 years, I get nervous. So then I'll grow it up again and refreeze a new batch. But yeah, I know some fungi, even in the refrigerator, there's a special way you can get fungi to go dormant under sterile water. And one of my colleagues at Michigan Tech, he revived them after 20 years. Just in the fridge. So that's pretty darn good. So a freezer, I usually put them at -80. That's burn your finger. I guess it's burned, right? I mean, it damages your fingers if you touch -80 stuff. Metal especially. Okay. Here's another technique that can be used. And there's actually 2 versions of transduction. There's generalized and specialized. So this one is the most complicated because you have a middle player in the game. You have 2 bacteria plus a virus. So what you have here is the first host. So we're going to learn more about viruses, but for this, I think you might have a basic understanding with everything going on in our culture and currently that the viruses, you know, from the virus perspective, they want to take control of that host cell so that they can make more copies of themselves. That's their goal. Okay, so that's what you see happening here. You have this bacteria, here's the chromosome of the bacteria. And viruses do have nucleic acid inside them. Here you see it's purple, but it is getting injected into the host cell. Now, normally, what happens is the virus goes into its replication phase where it takes that cell hostage and it says, make more of me, viral particles, many, get going. And so what tends to happen as that is occurring is the host DNA fragments into little chunks. Okay. Now you're seeing the replication going on, the nucleic acid inside the virus is directing, make more parts of the virus, and then they start assembling it all together. And as they're doing that, they have to package in their purple viral nucleic acid. But you see here, that's not purple, right? Great. So what they're trying to show here is that every once in a while, the viral particle gets the wrong DNA put into it. It's host DNA that's getting put into it instead of the viral DNA. So then, after all the replication is done, what you see in the last image here, you see that way they drew it here, it's a little lopsided. More of the viral particles have host DNA. I think it's actually the other way around, where it's mostly, you know, correct viral particles, but then a few that have the host DNA accidentally packaged in there. Now, for the first host cell, it's doomed because this host cell is going to break open, lyse. All these virus particles spew out. And their goal is, I need to find another cell and infect. So this first host cell is doomed, but the second one might just make it. So what's going to happen is after this first host dies and those viral particles get released. Now imagine a neighboring bacteria right there. And the virus that has this this bacteria DNA from the first host could go to that nearby cell. So that's what they're showing here. So this first host is toast. Right. And so now this virus particle finds a neighbor bacteria and injects some of that DNA from the first host. Now this bacteria could say, hey, I'll take advantage of that, and they might paste it into their chromosome, and it might be useful for the second host. The other key thing that tends to happen is this viral particle is defective. So it can't infect the cell, it doesn't have enough of its own genes to do that. And so it gets the DNA injected in, but that's pretty much as far as it goes. It can't take that cell hostage. So the second host bacteria could get a good advantage here in this transduction experience. So the movement of DNA from the first bacteria to the second bacteria, and this process that you see here, this is the generalized transduction. We mean generalized because when this virus is making copies of itself, this DNA is all fragmented. Everything's kind of just floating around. And so it's like totally random which chunk of DNA might get put in the virus. So it's generalized. Who knows? Just some section of the DNA. So it's a very general process, if that makes sense. So that's why they call it generalized transduction. Next time we'll talk about specialized transduction, which is a little bit more targeted, which piece of DNA is actually going to get brought into the virus. Okay, but we'll stop there. Any questions on generalized transduction? Okay, we'll recap this on Friday, and Friday, I think I'm also going to try to do a Google Forms thing with you in class. So let's give that a shot on Friday and see if it works.
So